thank you very much for inviting me and organizing this event. It's a pleasure after 2 years of no physical speaking. And today I'm going to talk about some work. It's a work in progress that I'm doing during my postdoc with Amindus Mohammadi and Roman Müller. And essentially I'll be presenting a phasefield model which we use to model the behavior of cell monolayers. So the key interest in our group is cell migration. It is governed by multiple genetic, chemical, and mechanical factors, and still not very well understood. It takes an important role in various different biological processes, such as development, wound healing. It is present in pathological conditions, such as cancer metastasis. And it's also a thing that happens in vitro experiments. Even though all these examples are very different and occur at different stages and organisms, there are some general features that are present. And one way to approach and model this general behavior is to make a connection with liquid crystals. So these are something between solids and liquids. These materials have orientational order, but no positional order, so no crystalline structures. And they can be subclassified further, and one type of liquid crystals are nematic liquid crystals. So essentially these are elongated rods that in certain conditions align in a given direction to create a nematic order. This is shown on a diagram. And if we think about biological entities, either bacteria or cell, they can also be elongated, but the only difference with biological entities is that they are active in the sense that they take the energy from the environment and convert it into mechanical work, and they move. So we call the structures that we work as active nematics. And we have seen experimentally that indeed biological tissue represent the key features of nematics. So they have order. They also have topological defects, so these are singularities in the orientation of the rods. And also, one example would be bacteria. They are elongated, so this is quite obvious. Similarly with fibroblasts, they also have slightly elongated shape, but we also see the features of active nematics in epithelial cells, which are normally quite circular, have circular areas. And when we active nematics has several features. So the activity that is present in these entities, it destroys this long-range nematic order when everything is lined in the same direction, and leads the system to so-called active turbulence, where we encounter a very chaotic flows with swirls and jets. And during active turbulence, we also observe continuous formation and annihilation of topological defects. And in this talk, I'm mainly talking about 2D materials, so 2D active nematics, and the most common topological defects in the system are plus halves, and so they're comet-like in shape, and minus halves. They're trefoil-like in shape. These two defects come in pair, and once they're generated, we observe experimentally and from continuum liquid crystal theory that trefoil defects, minus half, is usually passive. It does not move much, whereas the comet-like, they have self-propulsion, they move, and the direction of movement will depend whether on the type of activity you have, whether you have extensile stresses, so the particle pushes the material along its elongation axis, or it has contractile stresses, so it pulls the fluid towards its elongation axis. So if you have an extensile system, then the comet will move from tail to head, and if you have contractile system, then it will move from head to tail. And topological defects, surprisingly, are important. Also, they have a biological function. They have recently few experimental studies where they show that topological defects can control cell death, extrusion. My favorite example is an experiment where they looked at the small animal hydra during development, and they observed that where you have a topological defect will later be a limb forming. And however, we still don't, we observe this nematic behavior in the systems, but we don't really know how to bridge the microscale behavior with the macroscale. So why we observe this nematic features. And we can approach this theoretically, and there are a few methods that we can use. The most common is to use continuum liquid crystal theories, adaptive for active systems. But such models do not incorporate any cell heterogeneity. We don't have information of what happens at the boundaries of the cells. Another way would be to use self-propelled point particles models, but these are not, these are usually for the systems that are far from dilute limit. And we have used instead a phasefield modeling approach where we model cells as deformable active droplets. In this model, we can then know exactly what happens at the interface of the cell. We can calculate all the forces. And we know that this model has been successfully implemented also to model cell monolayers. But before, most of the work assumed that this model, these cells, they have polar driving, so they have certain orientation where they want to go. Not much work focused on the modeling the contractile activity. And recent experiment, we are motivated by recent experiments that show that even though cells are usually assumed to have extensile activities, in some cases they can switch to contractile activity. For example, when you knock out E-cadherin, the cells will show contractile activity, and they will also demonstrate more metastatic features. So we find it is very interesting to understand what leads to this switch and what consequences it has for the cell movement. So we have developed a phasefield model where we have a cell and we define for each cell phasefield phi I, and it can take two values which denotes the interior of the cell if phi is 1, and exterior of the cell if it's phi is 0. And the dynamics of the phasefield is governed by this equation, and in it we have an advection term with the velocity yi, and it acts on the cell as a whole without deforming the interface. And we have a functional derivative of free energy, which sets the interface itself. And so, since we're modeling a monolayer, we have a lot of these equations for each of the cells. So our free energy has three contributions. The first one is Cahn-Hilliard free energy, which has two terms. The second one is the mixing part, and the first one is demixing free energy. Essentially, it's a double well potential with two minima. For each of the phases, you have phi 1, which is interior, and phi equals 0 exterior, and the boundary between the two interfaces occurs exactly in the middle at phi equals 0.5. And if we assume steady-state problem, we can obtain a solution for the interface, and on the second plot, we just plot the interface for different values of lambda, which is the interface width. So we can vary the parameter and have this. So then the other two terms in the free energy first is the area constraint. We make an assumption that cells should keep more or less circular areas. And we also make an assumption that the area is not converged, so the cells can be squished or stretched, which is more biologically accurate, we think. And the final term is a repulsion term, so it penalizes the overlap between the cells. So then we having defined the interface of the cell, we couple it to the hydrodynamics. So we assume an overdamped system where we have force balance equation. So we have substrate friction on one side and the forces at the cell interface on the other side. And we incorporate the forces at the interface using stress tensor sigma. And since we want to see the nematic behavior in our model, we include dipolar interactions into these stress tensor. So we define a nematic tensor, which is essentially the orientation of the cell, as Q. So the Qi is for a single cell and Q is for the whole tissue. So then we write the tissue tensor as it has two terms. It has pressure term, so pressure from the neighboring cells, and it has this second dipolar force density distributed along the interface, which is essentially the activity term. Zeta is the activity parameter, and it can be either positive, modeling extensile activity, and negative, having the contractile activity. And this term is exactly what we have in a continuum theory, so it makes it very easy to then relate the two models and make a comparison. And finally, we also have an equation for the dynamics of this orientation, so the nematic tensor, and we define it using the angle of the orientation. And in active nematics, most of the dynamics occurs from the elastic restoring force and the flow alignment. So the orientation tries to align with the flow. And we incorporate this into our model by introducing two torques, so k tau and j times the omega, which essentially mirror what we have in the continuum theory. So then we do numerical simulations. First of all, we want to validate the model and make sure that it correctly reproduces the behaviors we see in the continuum liquid crystal theory. And here is, we start the simulations by placing cells randomly on a 2D grid. So here on the plots on the left we have cells, phases, and on the right we have coarse-grained nematic director. And you can see that as you run the simulations longer, you start, the active turbulence occurs and you start having topological defects, which are denoted with squares and triangles. So they move, interact, annihilate each other, and proliferate. So we quantify all of these behaviors that we observe. And first, by starting at the beginning, we know that the activity, when nematic is active, it develops bend-splay instabilities. So you can see this on the first two plots, that they start to develop this walls of similar orientation, and we define the measures to calculate bend and splay. And here are the plots where we show that from the beginning of simulation, after a certain time, you will develop non-negative, non-zero bend and splay for both extensile and contractile activities. So then we also did the linear stability analysis, so we made some very simplified assumptions to derive a continuum approximations of our phasefield model. So we assume that the system is passive, so the cells take hexagonal shapes. We also assume that the cell width, the width of the cell interface is very small compared to the cell size, and we were able to derive a critical activity, so a value after which we will start seeing the bend-splay instability. So if activity is below, the system will remain passive and there will be no movement, whereas it is above this critical activity, then we'll start seeing bend-splay, and if we go further, we'll have active turbulent state. And this is just to demonstrate this onset of creation of the first pair of topological defects. So you have these walls of elongation in the nematic field, and along this wall you see a formation of pair of topological defects. And as expected, we see that the negative, so the threefold defect, remains more or less in the same place, so it's passive, whereas the positive defect, it travels along the wall further. And we show that the time it takes for the system to develop the first pair of topological defects decays exponentially with the activity, which is consistent with the continuum theory. And then we move on to looking at the active turbulent state, so a dynamic state, and we look at the dynamics of these defects. So we calculated the rate of defect proliferation and also density of defects as a function of activity. And as expected, we see that as you increase the activity of the cells, then you obtain more and more defects until these values of rate and density saturate. But what was interesting is when we tried to look at how motile the defects are. So we measured the velocities of the defects, and as I mentioned before, from continuum theory, we know that plus halves should be motile, minus halves should be passive. And we observe this at low activities. However, if we consider more active systems, in this case, then both types of defects obtain the similar velocities. So they become diffusive. And it is in contradiction with the continuum theory, but it agrees with other phase field modeling that has been done. And we think this is a feature of the models that model cells as active droplets, and this behavior is a result of a defect-defect interaction at longer times. And also there are some experimental data that on human bronchial cells that also observed the subdiffusive motion of plus halves and minus halves. So this is something that we're planning to investigate in more detail. And if you're looking at the averaged properties of the defects, so we took average director fields and velocity fields for both extensile and contractile activities. So for director field, we can correctly capture the shape of the defect. So we have comet and trefoil. And also for velocities, for the plus half, you can see that you can have a switch of the movement, whether it is from head to tail or tail to head for extensile and contractile activities. And then another check that we did was to calculate the velocities of the tissue. We know that we have a periodic boundary condition and a confluent monolayer. Thus, the total force should be approximately 0, so there should be no velocity developing. And this is what we see in the first plots on the dashed line. So the total velocity remains 0, which is expected. And we also we calculated the divergence. And in continuum theories, the major assumption is that the cell monolayer is incompressible. However, if we increase activity, we see that our monolayer starts developing non-zero divergence, and so it deviates from incompressible behavior. Yeah. And finally, what we observed with our model is spontaneous formation of gaps, which was a bit unexpected. And so here you see two examples of two simulations. So on the left-hand side, you see a confluent monolayer, and cells move without creating any gaps in between. On average, the tissue density is quite homogeneous, but then in certain parameter regimes, the picture is very different. The density of cells is quite heterogeneous, it's quite active. You start having these gaps forming, so there are no cells. And we saw that this behavior appears when the activity is high. So if you increase the activity of cells, the gap starts forming, and you can see this from the plots on the top, where we measured the fraction of free space in the simulation domain, which increases as you increase zeta. And another interesting observation was that it also affects the overall shape of the cells. So if you have no gaps, the monolayer is confluent, then the cells have more or less hexagonal shapes. However, when the gaps are present, then the cellular shape tends to be more circular. And as you increase also activity, you will observe more heterogeneity in cell shapes. And this is also something that we're going to pursue to explore more, because we have some experimental collaborators that also actually observe this type of gap formation in one of the cancer cell lines. So to summarize, we have developed a phasefield model. Our idea was to develop the most minimalistic model in order to capture this nematic behavior of cells, so we can try to link what happens on the microscopic level with the microscopic features. We were able with our model to correctly reproduce bend-splay instabilities, formation of topological defects, active turbulence, and also we saw the formation of gaps, which we're going to investigate further. And currently, what I'm working now is that we are using the model to study the competition between extensile and contractile cells. So we want to see how they interact with each other and what kind of behaviors it leads to in a context of epithelial to mesenchymal transition in cancer. So the switch from non-motile to motile, more metastatic conditions. Thank you very much.
In your last pictures on the gaps, is there any correlation between the positions where the gaps appear and the defects? I mean, are they colocalizing with the defects or not? And
we did a correlation, autocorrelation functions, but we didn't see much information about that, unfortunately.
I have a question. What are the cells which are extensile?
They're the majority of cells are.
What cells?
Extensile?
Yes.
It's just how they move. So they push the fluid along their elongation axis. So they're doing these things.
And do you know the name of the cell?
The majority of cells are extensile, like majority of types. They demonstrate on the they have a contractile forces, but the flow patterns they generate resemble extensile activity.
Yeah, thank you for your talk. I was wondering, there are some experimental evidence that show that anisotropic cells can have a differential friction in their direction and in the orthogonal direction.
They are.
Do you have asymmetric friction? And do you know if what it would have as an impact?
We don't have it in this model, so I cannot predict what has happened. Not in this model, no.
Any other questions?
Thank you for the very nice talk. Besides the parameter zeta, what are the other parameters that control the formation of those gaps?
It's elasticity.
Okay.
So how elastic the cells are, so whether they interact elastically with each other or not.
Okay. And is there any interplay between elasticity and the parameter zeta?
No.
Okay.
No.
Okay.
They appear in separate places.
Okay, thank you.
Okay, I think we can thank again Aleksandra for the presentation.